Is the absence of water. This is going to inhibit metabolism. Microbes can remain viable and dormant for many years in this. The effects again can vary based on the microbe. So some might be affected more by desiccation. They dry out and they no longer can affect you. Others can last longer due to like an encapsulation or even the endospore that's present. So when we look at these examples, the Neisseria gonorrhea, this one can last for about 1 hour after it starts to dry out, whereas the tuberculosis can last for months and endospores can last for years. We also then see osmotic pressure changes. This is achieved by hypertonic environments. This could be with high salts and sugars. This causes plasmolysis. This is used a lot of times when we talk about canning or even preserving foods. Like with salts, it would be more for like meats. When we look at sugars, that's more for your fruits. This is one reason why you'll see like if they're put in the little jars and stuff like that, they contain the high sugar, and it's to help decrease the growth of microorganisms. And the point here is to inhibit their metabolism as well. The next main method here is radiation. When we're looking at radiation, we can use ionizing radiation or non-ionizing radiation, but it doesn't matter which one you're looking at. Both of them do affect the DNA. Ionizing radiation, though, destroys the DNA, whereas non-ionizing just damages it. Okay, and so this is what we're looking at. Now, ionizing is not widespread in routine sterilization, but they do use it to sterilize some pharmaceuticals, medical, and dental supplies. The non-ionizing is the radiation is not very penetrating. That's one reason it's not used, but this is to control closed environments with like UV lamps, and this is more germicidal, all right, and so it's to kill the germs that are present. So with radiation, it can kill microbes depending on the wavelength, the intensity, and the duration that it's exposed to this radiation. When we look at ionizing radiation, this is going to be the radiation that has the higher intensity, the shorter wavelength. These are going to include the gamma rays and the X-rays. They can be used to sterilize heat-liable drugs and medical equipment, but the reason for this is they do alter the DNA. This is why you should not expose yourself to gamma rays and X-rays. If you get X-rays occasionally, it's not that big a deal, but that person who is the X-ray technician, they need to protect themselves if they're giving hundreds of X-rays a day. All right, and so these are the ionizing. The non-ionizing would be more like the UV lamps. They're there to damage the DNA. They produce a pyridamine dimer, where we see that if there is a thymine next to a thymine in the DNA strand, it actually causes them to pair with each other instead of pairing with A like they're supposed to. The UV light is good for surface decontamination, and so this is what we see, and this is one reason why UV radiation does potentially cause skin cancer for us too, because it does damage our DNA, and in the case here we would use it to damage the microbes' DNA. Another type of the non-ionizing radiation are microwaves. Microwaves have a very low energy wave. It does not have a direct effect on the microbes, but the high temperature from the water does. When we microwave food and things like that, we're actually heating up the water molecules that are found in those substances. This is what causes that heat to happen, and that's one reason why it's really uneven when you have that heating and you have to kind of stir it up. This will also start to affect the microbes too, based on how much water is present. All right, now switching gears, we looked at the physical methods. Now let's look at the chemical methods. Chemicals can reduce bacterial populations or remove vegetative forms. A lot of times they're non-endospore forms. They're not going to affect the endospores. These generally don't achieve sterility. Unlike what we saw with the autoclave and things like that, we're not going to actually sterilize this. There are different disinfectants that are effective for different microbial groups. We find that some things like bleach will work better for some microorganisms, but not others. All right, so the first group of chemical agents we want to look at are the phenols. Okay, when we look at this, you have the phenols, the phenolics, and the bisphenols. When we look here, the phenols, all of these are going to look at disruption of the plasma membrane. They also are going to help with denaturation of enzymes. Now guys, phenols are rarely used except as a standard of comparison. These are seldom used as a disinfectant or antiseptic because of its irritating quality, like on the skin, and it has a really bad odor to it. The phenolics are going to be for environmental surfaces like instruments, some skin surfaces, viruses, and mucous membranes. This is a derivative of phenol that are that are active even in the presence of organic materials. Bisphenols are going to disinfect. They're going to be disinfectant hand soaps and skin lotions. Triclosan is especially common example of this. It's a broad spectrum and it's most effective against gram-positive bacteria. So guys, when we look at these groups, phenol was actually used by Lister. And if you remember when we talked about him in Unit 1, he was the first one that started disinfecting. And he was the one that was trying to reduce the idea of the childbirth fevers and also during surgeries and passing on infections. However, he found that this is very irritating to the skin and it did have a really bad odor. This then led again to the phenolics. This is not as irritating to the skin. One of the main groups is what we call cresol. It's the main ingredient in the disinfectants like Lysol. And then the bisphenols are like the triclosan. This is in antibacterial soaps. We also have the hexachlorophene. This is found in some prescription or surgical scrubs. And this is especially effective again against the gram-positive bacteria. We see triclosan, if you look in these pictures, can also be used in toothpaste. All right, so if you look here in the toothpaste, you can see Colgate has that, whereas Crest does not. One issue with using the triclosan too much is if it can cause widespread. If it's used in a widespread way, it can generate resistance of bacteria. So we have to kind of be careful on that use as well. Now, in order to determine if these are present, you have to look at the label. And this is again why labels are located here on this slide. So the next group are the biguanides, also known as the chlorhexidines. These are going to disrupt the plasma membrane. We can use these for skin disinfection, especially for surgical scrubs. These are bactericidal to gram-positives and gram-negatives. These are non-toxic. And they are persistent. And so when we look here, some examples, this is showing you the antiseptic type of mouthwash, and we also have some other cleaners. And again, you would be looking for that chlorhexidine on the label. The next group are the halogens. The halogens are effective against all kinds of bacteria. They also are effective against their endospores, viruses, and fungi. The main thing that these guys do is they're going to help inhibit the protein function. There are two main forms here. The iodine is going to be used as an antiseptic, and the chlorine is going to be used as a disinfectant. So recall earlier in this lecture, an antiseptic means it's going to be used on living tissue, and a disinfectant is going to be used on inanimate objects. All right, so this is what we're looking at. Now, this first particular slide, it's showing you the iodine products. All right, so the first one here is the povidone. The povidone is a lot of times used in first aid, and the whole point is to decrease wound infection. We also see betadine. Betadine is also used in wound disinfection. It also can be, when we look at iodine especially, these could be used to treat water. Campers sometimes use this to help treat the water before they drink it. The last one here is the tincture. This is do not stain, and it's less irritating to the skin than the other types of iodine. It's put into an aqueous type of alcohol solution, and it is released more slowly. Chlorine products are a little different. We have the bleach. The bleach has sodium hypochlorite in it. We also see the chloramines. This is chlorine in combination with ammonia. One thing with this group too is it doesn't leave a residue, and it's odorless and tasteless. The next group are the alcohols. These include things like ethanol and isopropanol. These are going to do protein denaturation and also lipid dissolution, meaning that it's going to dissolve the lipid components, the fats. We use these a lot of times with thermometers and other instruments. We do this also to swab the skin with alcohol before we give a shot or something like that. And the whole point there, though, is it's simply wiping it away. And so it's more of a degerming than it is an actual like sterilization. This is bactericidal and fungicidal, but not effective against endospores or enveloped viruses. And so again, we can use this. This is rubbing alcohol. This is less volatile than just normal alcohol, and this is why they have a different percentage to them. Now, these percentages are kind of important because we find that if the percentage is too low, like 40 or even 50%, we see that the growth still happens with a lot of these types of bacteria. No growth starts to happen at 50% if it's been left on for 30 seconds or longer. If you look here also at the 100%, if you use 100% ethanol, there's growth with the time of exposure. And so 70% has been found to be the most effective percentage for denaturing the proteins. And so this is why most of these products on the market are normally a 70% ethanol concentration. And the reason this percentage is less than 100% is that in order for denaturation to take place in the proteins, water has to be present. And if it's 100% ethanol, there's no water that's present. All right, the next group we're looking at are the heavy metals. Heavy metals are can be used in very small amounts, and they can kill or inhibit growth. The way they do this is they denature enzymes and other essential proteins. Heavy metals such as silver and mercury are biocidal. So when we look at this, some examples are things like silver nitrate, muric chloride, and copper sulfate. And so these heavy metals help in, again, inhibiting or killing the bacteria. Some heavy metals have antimicrobial effects. So if you look here in this example where they put coins that had different metal concentrations on the bacteria or on the spread plate, you can see that some of them killed or at least inhibited the growth, whereas others didn't. Inoculating loops are made of platinum, tungsten, or nichrome. So this way it does not kill the bacteria, because if you use something that had silver, copper, or something like that, it would kill the bacteria before you could transfer it over. Now, when we look at this, guys, the mercuric chloride is very toxic. It's corrosive. And so it's ineffective on organic materials. The chloride, though, also tells you that it has a kind of bacterial static type of structure or a role. And so this would be used more as a disinfectant. Thimerosal is an example of an organomercury compound. However, this is, and you can see it here on this label. However, newer products on the market are actually mercury free, even though they kind of show that they seem to have mercury in them. They're actually mercury free, and you can look at this on the label because there's no mercury contents, and they actually contain a different type of chemical that's called a quat, which we'll talk about in a minute. On the other hand, there are many different things that use silver that are on the market. One is the silver self-design. This one is going, this is going to be used a lot of times on burns. We also see that you can use silver nitrate solutions. These are used as a prophylaxis on babies. If you'll notice, once babies are born, they put these eye drops in their eyes, and the whole point is to help be proactive in making sure that there's no infections of the eyes. Especially when they go through that birth canal, there's a chance that they can be exposed to lots of different types of bacteria or viruses, and so we want to potentially protect the eyes. Now, silver has actually got an increase in its interest because we do find that it can work against antibiotic resistant bacteria, so potentially coming up with types of medications that have silver in them that can be used against those instead of antibiotics might be helpful. Now we want to look at the surface actives. They're also known as surfactants. These are things like your soaps and detergents, and also the quats. The quats are what we call quaternary ammonium compounds. When we look at soaps and detergents, this is more of a mechanical removal of the microbes. This is more through scrubbing. It causes skin degerming and removal of debris. The quats are going to do enzyme inhibition, protein denaturation, and disruption of plasma membranes. It could be used as an antiseptic for skin, instruments, utensils, or even rubber goods that cannot necessarily be heated and things like that. So let's look at soaps and detergents first. These are what we consider emulsifiers. They do not kill or inhibit the growth of the microbes, but they mechanically remove the germs from your hands or from the surface through scrubbing. It breaks the film of oil, dead skin cells, sweat, and microbes into droplets, and this is what's called emulsification. And then we can rinse them away with water. When you rinse them with water, it lifts and carries away those droplets, thus de-germing the skin's surface. The next group are the quats. These are the quaternary ammonium compounds. This is the most widely used type of surfactants. They have positive charged molecules, which gives the quats their cleansing ability. They actually are really good against killing gram-positive bacteria, fungi, amoebas, and enveloped viruses. However, they are not good against endospores or mycobacteria. They do not take care of those. Again, looking at these labels, you can find them as the active ingredients. When you look at Scope or a mouthwash, when you put it in your mouth and you start swishing it around, it causes that foaming action. That is due to the quat that's there, that's listed here. This is also known as Cepracol. We then see that we can see some others here in these example of these antiseptic towelettes. This is also known as Zephyrin. This can help again with cleaning off surfaces. The good thing about quats is they are colorless, odorless, tasteless. They are very stable, and they're easily diluted in order to be used in cleaning supplies, as well as being non-toxic. The next group are the aldehydes. The aldehydes include formalin and glutaryl aldehyde. These are going to do protein